అండి మన వాళ్ళకి వినిపిస్తుంది म्यूट हो गया ఇప్పుడు మ్యూట్ చేశారు ఇందాక అన్మ్యూట్ లో ఉంది మాట
उडुपा सर नमस्कार उडुपा सर नमस्कारी उडुपा सर नमस्कार hope you are able to hear me.
Yes, I can hear you.
I am talking to Udupa sir. Hello.
Hello, good morning. We will now start. Good morning all, good morning once again. This is your host Jojo and I welcome you all to this free webinar on pharmaceutical technology this February 5th of the year 2022, organized by D.Y. Patel University School of Pharmacy and Association of Pharmaceutical Research and our scientific partner BioLeagues Worldwide and research partner United Innovators. About D.Y. Patel University School of Pharmacy, D.Y. Patel deemed to be University School of Pharmacy is an institute where we shape specialist pharmacists well equipped to pave a way towards progressive healthcare. It is our earnest effort in creating individuals who will fortify the healthcare system by working alongside healthcare practitioners. Our excellent faculty discounts no efforts in making the students able pharmacists who will find career opportunities in community pharmacies, hospitals, universities, government agencies, and etc. We believe in providing practical education in addition to the theoretical study practices to make our students well equipped to handle real life scenarios. Our university's futuristic healthcare model is an ideal place for an aspiring pharmacist to gain experiential training from the most accomplished professors in the industry. A little bit about the Association of Pharmaceutical Research. Association of Pharmaceutical Research is one of the world's leading professional association caring for people. APR is non-profitable professional association meant for research and development in the field of pharmaceutical science and technology. APR is an international forum for researchers, academicians, doctors, and practitioners for sharing knowledge and innovation in the field of pharmaceutical science and technology. APR aims to bring together worldwide researchers and professionals, encourage intellectual development, and providing opportunities for networking and collaboration. APR meets with its objectives through academic networking, meetings, conferences, projects, research publications, academic awards, and scholarships. APR also strives to enrich from its diverse group of advisory members. APR brings together pharmacologists, life scientists, pharmaceutical practitioners, and researchers to advance the application of pharmaceutical science and technology to the public. APR delivers access to its members and the industry's most essential technical information by organizing conferences, workshops, annual convention meeting, and provides networking opportunities both locally and globally. Members have the ability to stay current in their chosen profession, connect with peers, and of course, invest in their future. A little bit about BioLeagues Worldwide. BioLeagues Worldwide is a not-for-profit professional association which prominently promotes research and development. We at BioLeagues Worldwide have brought a revolution In the field of worldwide conferences, BioLeague's worldwide conferences bring together the professional wizards and leaders who have explored all avenues to reinforce the field of life sciences and medical technology. BioLeagues Worldwide conduct events worldwide which help in enhancing the skill set of the people from diverse industries and also forms a common platform for eminent personalities, physicians, researchers, doctors, and academicians, professionals, business figures, and many more. BioLeague's conference encourages better comprehension about improvements and progressions over the world through worldwide conferences with the speed of science and technology. We work with our motto of creating a better tomorrow by organizing conferences and creating a network which will help grow a better tomorrow with the help of advanced technology and achieve sustainable development. Now, I would like to welcome Mr. Rudra Banu Satpathy, CEO and founder, Association of Pharmaceutical Research or APR, a unit of Technorit Group, India. Welcome, sir. Good morning. Sir, you're on mute. All right. Seems like internet technical problems. Let us welcome our first speaker for today, Dr. Rakesh Sumani, Principal, D.Y. Patil University, School of Pharmacy, India. Sir, good morning. Welcome.
Yeah, good morning. Thank you so much. Yeah, it gives me an immense pleasure to welcome all the dignitaries and dear students and faculty members who are present on board today to attend this very, very informative scientific session. I'm really indebted to the team APR, particularly Mr. Rudra Sarpati, the founder, for organizing this particular event with us. And D.Y. Patil University School of Pharmacy, Navi Mumbai, is one of the institutions which have come of late past 3 years in the Mumbai campus. However, we have other institutions which are located at Pune, and they are there into education field past 20 plus years. This brand, the D.Y. Patil University is itself has been established as the brand for the imparting the education, quality education, I must mention. I'm really happy that APR and the D.Y. Patil University School of Pharmacy are collaborating, and we look forward for such kind of active seminars, webinars, conferences, workshops, and a lot of scientific exchange on this platform henceforth. I'm also happy to see Professor Udupa and other speakers over here today, and really looking forward for a great intellectual discussions between the students and the speakers. Thank you so much for giving me an opportunity to talk here.
Thank you so much, Doctor. Next, I would like to welcome our first speaker for today, Dr. Hashim Ahmed, PhD, WHO Fellow, Director of Research and Development, Center for Biomedical Research, Population Council, New York. Sir, good morning, or I think good evening. Welcome. You're on mute, sir.
Can you hear me now?
Yes,
Yeah.
I can hear you now.
Yeah, I was
clearly.
trying to speak and it gave me a message that the host is not allowing me to unmute. So
Oh, my apologies. My apologies.
yeah, it's all right.
You are now. We can now hear you clearly. Go
Exactly.
ahead, sir,
Thank you so much. Yeah, I was there. Thank you so much for having me to the well, what time? I don't know what it is now. It is almost 12:30 midnight in New
This is
York.
the-
Actually, not New York. Actually, I'm calling today from North Carolina, south of
Oh.
United States. And my name, as you said, Hashim Ahmed, and I'm really, I think you introduced me nicely. Thank you so much. Thanks for Ramia Divai, who actually was also asking me to join. So actually today I'm going just to share my screen. Let's see if I could do that. Yeah, hopefully it will
Go
work
ahead.
out. Yeah. Yeah. Okay. My presentation is here. Let me see if I could do that. 1 second. Sorry guys for that. Look like, because usually I use the Meet system, but let's see if this, yeah, if this, yeah, let's see. How can I move this? That you're here, yeah. I don't see my files. Let's see. That is strange. The screen doesn't come. 1 second. Let me try one more time.
Yep. On our Zoom meeting, you will see there a share screen green button just right below of our
Yeah.
photos.
That's what I'm using. I'm
Okay,
familiar
when you
with.
click on share screen, you will see tiles of windows that you will choose. Make sure to choose your presentation before you click on share. There's another share button right below.
Yeah, I just hit the first one, which is share screen, right?
Yes. Then there will be a window that will come up, and please choose. There you go. Yep. We do
Yeah,
see
but
your screen. You can go ahead and click on your
yeah,
presentation.
but you see it?
No, I'm seeing just your
1 second. Let
your
me see.
meet.
Get out of this one. Sorry, guys. Very bad technical thing. Okay, let me see if I could. 1 second. Bookmark. Okay. Why is that?
Is your presentation open?
Yeah, let's see. Yeah, presentation is open. Yeah, ready to
Okay.
go.
I will stop sharing your screen again. Then
Mhm.
please share your screen again. When you click on
Yeah.
share screen below.
Yeah. Now it works. I think. Yeah, you should see it now, I guess.
Yes, it is. Yes,
Yeah.
you may proceed, doctor, please.
Something was, yeah, competing with. It's okay, no problem. 1 second. Let me just move this here. 1 second. It's a lot of technical stuff here. All right, that's good. Okay, sorry for that. So it took some minutes from me, but yeah, thank you again for having me today. So I'm going to talk today about, the formulation business. And I think there are so many of our folks here are, formulators, analytical folks, etc. So I try to have this presentation in a way which will actually reflect probably my own kind of experience, but also the experience of, of the formulation, and also how the formulation and how the analytical and how the pharmaceutics business can help move the medicine and the development of the of our products throughout the different kind of steps or different kind of phases in development. So this is first slide. Actually, this is a pipeline. All the time people ask question, what is a pipeline? What's the drug delivery? Sorry, what's the drug development process? So in front of you, basically, this is how we develop medicines. Obviously, this is mostly the United States kind of experience, but it is universal, obviously. I think you guys are all familiar. Basically, you will have the target identification, and then when you are developing a medicine from the beginning, you guys are, School of Pharmacy, so most of the professors here teach this kind of courses, but this is more kind of a practical approach, as you will see, and I also have some case studies which I'm going to show with you, how the formulation, how the analytical, how the pharmaceutics part of the business will help, accelerate the delivery and accelerate the development. Like I said, It's actually a long process, a little bit of complex process, but very systematic process itself. As you see it from the beginning, as I said, you have the target identification, then you have the lead identification, and then the lead optimization. The lead means that you know the molecule. You will have so many molecules, you know, under development, but not all of them are going to go to market. Not all of them going to be medicine. Those are molecules. So there's the difference between a molecule and the medicine. Medicine means it is ready to go. It doesn't have tox issues. It doesn't have kind of side effects, etc. Or always there is side effect, but already clean. That's why you go into the preclinical stage, which is basically the animal testing, and you do all of what you have to do to make sure the medicine or actually your molecule is actually doesn't have tox issues. Then you go after that, you go into, you file your IND, which is you know, investigative new drug. You know, when you would like to investigate new drug, you have to have application for what we call it IND, and that when you go into phase one basically safety and tolerability. You do all the safety in human. Before that, in the preclinical, if my cursor can show the preclinical, you show actually the animal work. Once you go into the clinical phases, that means you are doing it into human. Okay, all the work. So phase one, safety tolerability. Phase two, at phase two, you typically from a formulation point of view, that when you actually optimize your formulation, you finalize, unlock, we call it the formulation. Phase three, that when you have it in more people, more subjects, or more kind of, you know, kind of humans. And then obviously you go to phase four, which basically that is even post-approval. It's very expensive process. It could go up to 300 some plus $1 million to develop one medicine to reach the market. And this just, I give it to you, and I will share those slides actually with you guys. I will send it to Rami and to you. Then you could share with the rest of the people who would like to have more details. This is a very detailed slide, but very important. It shows you, again, the journey of the drug development, the journey of the medicine throughout the pipeline, you know, process. So from the exploration, from the beginning, from the discovery, we call it the intermediate. So in the exploratory, which I just mentioned, you talk about the new medicine proposal, the target identification, and then you go into the lead. When they go into the intermediate stages, that when you have the lead identification, then you develop the lead itself, the lead optimization. What I mean by that, those are all many molecules. We do something, we call it developability. And I will show you exactly what I mean by that in the next slide. But you try to make sure the molecule you have selected is well characterized, well developed. That's where actually the School of Pharmacy will have a big role here, because most of what you guys studies, we applied here. Okay, so you make sure that the drug has the right log P, log D, the right solubility, the right, you know, kind of, you know, the molecule itself, the structure itself. Make sure that it is some molecule which can be used in human. Then you go more advanced when you go into the clinical stages, which I just talked about. Then you go up to registration. When you register it with the agency, that means this is a drug, this molecule now change into a medicine, that's a product which has all of the attributes, it's ready to go for sale. Rest of it, a lot of details here, we call them milestones. When you develop your product, actually you have like so many milestones. At each milestone, there will be certain study, there'll be certain kind of requirements to make sure that medicine is, you know, safe, most important, efficacious, and also scalable, stable, and also has the right bioavailability. And obviously, you file at the end, you file what we call an NDA, new drug application, that will take you into to take your drug to the market. When they approve your drug, the NDA, you know, remember we start with IND, which is investigation in a new drug, but you end up with the NDA. NDA is the last stage of your regulatory hurdle. Okay, so basically you guys are professors and you students and others. So you guys are focusing two main area when you talk about formulation, when you talk about development. From a CMC point of view, chemistry, manufacturing, and controls, that's what CMC is all about. There are two main sectors, as you know, pharmaceutics. Of course, you guys here, School of Pharmacy, you have a pharmaceutics department and group, and also the analytics. So these are the two main sectors. And actually, you guys know exactly what it is. And, you know, Formulation. We always talk about formulation, but simply what the definition of the formulation is the transformation of the active ingredients into a real dosage form. You cannot give the active itself to patient, right? Can you? You cannot. So what do you do? Actually, you have to have the excipients, functional excipients, plus the APIs, the drug itself, and that combination will be the formulation. Of course, has to be a certain composition, certain kind of criteria, making sure the manufacturing process, the analytical testing is right. You develop the analytical method, you optimize the analytical method, you verify the analytical method, then you validate the analytical method, which will be testing to control. That's where the control will come. That will make it a drug product. Okay, what is the objective of the formulation? Why we do formulation, right? Because you would like to have the optimal delivery of the active therapeutic agent, and drug molecule to achieve the desired pharmacological effect. That's what the objective of the formulation. So make sure that you can maximize the exposure. Very important. Always about bioavailability. Always about exposure. Exposure means the AUC. That means you'll have the maximum possible AUC to get the effect. Because the medicine is not going to be efficacious. It's not going to work unless it's already in the blood, unless it has the right bioavailability, right exposure. Achieve the desired PK profile, which is, pharmacokinetics have to be right. You guys also study all these courses. Your professor will teach you that. We practice that, making medicine real medicine. That's the difference between working in the industry, working in other places. And also making sure that your dose is accurate. That means you optimize your dose. Ensure also your patient compliance, because you cannot give, for example, if you are dealing with an oral, you're not going to give the patient something which is very bitter. Then he's not going to take it. She's not going to take it. So the compliance is very important, convenience, and also acceptability of the medicine itself. If it is oral, it has to be like acceptable for the taste masking, we call it. And if it is like external, it should not cause irritation. It should not cause, any kind of side effect on the skin, basically irritation. So that's part of the patient compliance. Most important to have stable product, very important. So the role of the formulators, the role of the analytical folks, the role of the CMC, I call them, chemistry, manufacturing, and controls, function is discovery support. That means from the beginning, as I mentioned, basically selecting the optimal clinical candidate for the development. We talked about it. So from all those molecules, you do a lot of work to make sure you select the right molecule. Second, formulation for pharmacology and safety, the tox formulation might not be the same like the IND formulation, next one here, which is a formulation used to go into human. We call it IND formulation. Investigative new drug application, that is IND formulation. Development for the market formulation, that will be the final formulation. What is the difference between the IND formulation, the early formulation, and the market? The market will have all the attributes, right? The packaging and the know-how and how it looks going to the patient. That's the market formulation. The final formulation, the final product, the market product. Okay, so different kind of stages. Okay, so what are the key steps? Just quickly, physicochemical characterization. What these are the keys. Those are the steps when you develop a dosage form. Physicochemical character. First thing, you have to characterize your molecule. Select the route of administration. Is it going to be oral? Is it going to be injectable? Is it going to be topical? Is it going to be implant? Is it going to be an insert? What are you targeting? Depending on what the dose, depending on the therapeutic category, depending on many things, you decide what kind of a dosage form. Like I said, absorption optimization. You need, most important, to have exposure. So to have exposure, you would like to have absorption. Absorption could be oral absorption. Today, I'm going to talk a lot about oral, but it could be also my other area, which I'm expert on it, is dermatological. So you'd like to have enough medicine on the subcutaneous layer, and also actually in the dermal layer, and also in the, what do you call it, dermatological kind of percutaneous, absorption. Selection of the excipient, very important. Some of the excipient can be solubilizers, could be functional excipient, not just fillers. Selection of the dosage form technology, what are you going to do? Hot melt extrusion? Is it going to be tableting? Is it going to be injectable, etc.? Development analytical method, very important. You can't work blindly. You need to have the right optimized, verified analytical method. And also you have to consider all the commercial needs, and also manufacturability and stability. Here, the developability. We talked earlier about developability, and we talked earlier about, what are the the physiochemical properties, melting point, molecular weight, stability, intrinsic solubility in different physiological fluids, pKa, log D, right polymorph, also very important to select the right polymorph. What is polymorphism? Polymorphism is the existence of the same molecule in different physical configuration, okay, arrangement. And permeability, obviously, you can in orals or even percutaneous dermatology, you need to have permeable medicine to go into the physiological tissues, okay? Favorable for oral, just give an example, and to some extent also favorable for topical. Molecular weight always should be small one. Today we are talking mostly about small molecules. Maybe in the future I can teach you also or lecture or listen to you also about proteins. That's another area I work on, and large molecules. Today we are focused on the small molecules because we can't talk about everything in one presentation. So usually you would like to have a small molecule, less than 500 Dalton. That's the molecular weight. Solubility, more than 1 milligram. Permeability, more than 100. The scale of the Keiko2 cells. Keiko2 cell, which is kind of way of decide, what do you call it, assessing permeability. Melting point has to be a low melting point, because with this high melting point, it will be difficult to process. The role of the drug delivery scientist during discovery. We talked about it. So basically, you collaborate with the discovery team. So you, as a pharmacist, you are like, what do you call it, scientist with a pharmacy background. You will be very, very helpful in the industry, pharmaceutical industry. Talk about the United States. I work also in Qatar. I work in different countries. That is the same thing. Selection of the appropriate vehicle, buffer system. So a lot of basic pharmaceutics work is very useful and very important to help the discovery for, because you're working with pharmacologists, you're working with different folks. Other formulator, you'll be the what you call the milestone person who's actually helping to, you are the product guy. You make the product, right? Selected identification of the form, the salt, the ester, and also the physical and physicochemical properties characterization. As I said, you are the expert. Early this consideration of the delivery system you are doing, implant, injectable, like I said, oral, whatever, you decide on that one based on your experience. Here, I'm just showing this for an API, active pharmaceutical ingredient. What are the alerts? What do you have to look for when you have a medicine you want to develop? Maybe you guys have studied this one, but this is the industry, what we look for. Solubility, permeability, polymorph, and stability. We talked about solubility. Obviously, we know that in order to have a good solubility, in order to have a good molecule, and also which can be developable, you would like to have acceptable solubility, as I said, and also permeability, and also you don't want to have what we call it class 4. We'll show you in a minute what I mean by class 4, because that will be poor bioavailability, and also because of your solubility will be very low. Also very important, and focus here also in the alerts to make sure that you will have the right kind of properties. The polymorph, like I said, ensure you have the right crystalline form and also stable. It shouldn't be unstable, okay? Because if it's not stable, polymorph is going to change into different polymorph. I have so many case studies. I'll show you one of them, which caused problem to a company called Abbott, as you know it. Stability is very important. I mean, you can't work API which is not stable. It has to be stable. Oxidation could be a problem, hydrolysis, and all of those kind of factors you need to consider. Why polymorph screening? Because affects solubility, affect bioavailability, the polymorph, affect manufacturability, stability, hygroscopicity, right? So the polymorph, which you select has to be stable polymorph. If it is a crystalline, great. If not, there are ways of changing it from crystalline into amorphous. We'll talk about it in a minute. So you need to select the right polymorph from the beginning. Okay, this is a case, a medicine called Norvir. Maybe you know it, which is actually an HIV. And this Norvir was developed by Abbott, but they developed at that time, that was in July 27, it was a big case study in Abbott. And you could see here that the vice president of that company mentioned in 1998, they have an issue with the polymorphs. They couldn't develop the capsule. It's just because they select the wrong polymorph. So they couldn't do it because solubility was low. They couldn't improve it, so they have to use a solution. They couldn't do a capsule. Ritonavir. Ritonavir is a brand name, but actually basically, or ritonavir is is a generic name, and Norvir. In the market already, they fix it after that, but it costs them a lot of headache. Develop desirable, suitable delivery system for early clinical. So when you are in the clinical, now we talk about early development, the allergies, etc. Now you are going to clinic. You would like to go to human, clinic to human, phase 1, phase 2, and phase 3. So critical interaction between discovery and formulation, as always, analytical toxicology and a different function. Thoroughly explore your delivery options, because now you're going to humans. So you got to be careful. You're not going to go with very simple formulation, a little bit more complex formulation. Okay, so so entering to human, we call it IND, which is the clinic. The clinic, we call it the clinic formulation for the clinic. So formulation should allow wide dosing. So your formulation should not be like formulation which is you cannot change. It has to be flexible, because you still consider still phase 1, consider still early in development. It's not phase 2. Phase 2 is very critical. That's where you will lock the formulation. You optimize it, and that's it. You can't change it. If you change it, you have to have more studies to bridge. Whenever possible, employ simple form if you can. Don't make it complex. For phase 1, we are talking IND formulation. And usually you have to make sure that you target high exposure, like I said, as I mentioned earlier, because you would like to have a dose proportionality. So that means what we call it SAD, MADs, like single ascending dose, multiple ascending dose. So you will have a kind of you prove that when you increase the concentration, for example, you see your AUC increasing also. So that way you have dose proportionality. You need to prove that in the IND. So basically, and when you go to phase 2, that's not in this slide. When you go to phase 2, you would like to optimize your formulation, see what the issues you have with the formulation. If you have solubility, whatever we discussed, then you lock your formulation. You optimize it, because that formulation phase 2, typically we use it for efficacy, for gaining efficacy. Okay, phase 3 basically just to confirm phase 2, but phase 2 is the most important. Just remember that, and we can take question later why that and what do we do typically there. But basically optimization of the formulation, testing it into man again to get the efficacy. These are the 3 pillars, stability, bioavailability, manufacturability. If you're going to phase 1 to phase 2, whatever, especially phase 2 and beyond, you need to have a stable product, at least 2 years under ambient conditions in a conventional packaging. You don't want to baby it, we call it in the industry. You would like to have it in a conventional packaging. OHDP, that means those plastic bottles, or you may have it in blister packaging, whatever it is, but it has to be in conventional. That means not a complex kind of system, because it would cost the company money, and also would be difficult to reproduce. It's not a robust system. Also important, number 1 is stability, number 2 bioavailability. At least you would like to have 30% bioavailability for the oral, and also for the percutaneous bioavailability if you are dealing with dermatology. When you're dealing with injectable, might be a little bit more forgiving, a little bit complex in a different way, but forgiving in terms of bioavailability. Because when you inject, you go into we call it the bolus injection. Boom, it go, and your bioavailability would be almost like 90% and above. Okay, from the absolute bioavailability. So the oral is a problem because you have the gut, GIT, you have all of those kind of issues, and it will be more kind of complex. We'll touch on them. Manufacturability, of course, you can't go to market, you can't go to development unless you have a scalable, reproducible, and cost-effective. Also, I may add also. I'm showing here, some kind of technologies, and you have some challenges also. But basically, you salt formation, particle size reduction, lipid and microprecipitation. I'll give you some examples. These are challenges of bioavailability issues, etc. And these are the technologies probably we use to, overcome these issues. Before I go to that, oral absorption. Usually, for orals, if we talk about oral today, mostly, I'll talk about the principle of oral absorption, which is passive transport. So basically, this equation, which is J, is the flux actually across the GIT, across the gut, right? So it is a combination of the permeability and also the concentration profile at the gut wall. That means in order to have good flux, good movement, good flow of the active or your medicine, you need to have good permeability, you need to have concentration, higher concentration to give you what we call a supersaturation near the GIT, so that will give you more kind of osmotic pressure going into the system. And I will come to that later. But now let's talk about the factors which are very important for absorption, very important for bioavailability, very important for everything, which is the BCS classification system, permeability, solubility, permeability, and the classification. Class 1, which is the best, which we'll never find nowadays, because most of the molecules which we develop, they are very challenging. But if you get high permeability, high solubility, that's number 1. That's great. If you get low solubility, high permeability, you talk about class 2, we call it, not too bad. But if you're getting not too bad, actually bad, I'm not saying bad, but challenging also, but 3 is not too bad, right? But number 4 is the worst situation. If you have that, you have to have what we call it formulation intervention. You need to do more non-conventional intervention. I will give you some kind of, some idea about them. Okay, so if you have, the problem is when you have like class 4, this one, or class 2, these are like challenging. 4 is in particular, low solubility, low permeability. This one is low solubility, high permeability, which is 2. When the dose to solubility ratio is quite high, that means you have high dose and your solubility is quite low. That will be the worst situation. You will have lack of dose proportionality. You will have intra subject variability. You will have probably even side effects. So you want to actually to develop a formulation which can overcome this challenge of classification 2 and 4. One of the technologies we do is simply particle size reduction. And there are different technologies. Because of time, I did not put it all the technologies, but basically size reduction. Size reduction you could do by milling, you could do by ball milling, you could do by different kinds. There's a different technology, but typically by milling and using micronizers, right? So this is like air jet milling, and I have a whole presentation just for milling. If somebody interested, you'll have my email, and you could email me. I can send you different kind of milling we use in the industry. You could find in the books also. But what I will give you is what do we use in the pharma industry in the United States. So if you look here, because of size reduction, the solution went very fast. For example, when you have particle size like 2 micron, look at the dissolution. This guy is here, this one. Okay, so 2 micron, and then you start to have less and slower dissolution as your particle size start to increase. Okay, and we'll tell you, you must be knowing the reason. Basically, the surface area. When you reduce your particle size, you will have a large surface area available for your dissolution system, or in this case, for your even within the this is an in vivo studies, and it's very similar to this one, of course, in a different way. So if you have 2.7 micron, it's the same here. We moved here is in vitro dissolution, and this is the in vivo data. So it follows exactly the same way. So the significant improvement in the rate of solution, I'm saying, was increase in the surface area, decrease of the particle size, higher Cmax and higher agency. And that work, I did it in dog where I was at Roche, and the RO means Roche, that's CDKM, one of the oncology medicine we had. So basically we proved that just by milling, by doing the right milling, getting the right particle size, micronization. Micronization definition in the industry, less than 10 micron mean particle size. And this is just explanation of the theory behind it. You know it, very famous equation, modified Noyes-Whitney equation. Basically, the rate of the solution here is a function of the solubility and also the instantaneous solubility. Okay, so and this is thickness or kind of, you know, your what do you call it, your distance in your model itself. So basically, it's a function of the solution is rate is a function of solubility as well as saturation solubility as well as your instantaneous solubility. All right. So milling, you deagglomerate. The more you milling it, make it smaller as we have shown, you'll have better surface area, wider surface area, and also and also you will get better wettability. That means you will have better. There is no waterproof in between. There will be more wettability because they are very fine. There will be more kind of the solution system or even inside your GIT there will be much better kind of interaction with water, or actually the plasma. You'll have much better AUC. The same you could do it in a conventional way, which is I mentioned ball milling and all the milling and micronization. But you could do it in non-conventional way, basically what we call it by amorphizing the system, particle engineering, or you could do it by different ways. But my own experience was you do in situ size reduction by Amorphous technology. The drug will be changed into from crystalline into nanosystem Amorphous. That will give you an extensive surface area increase, which will give you much better bioavailability. Another example is basically, instead of doing that, maybe the salt. You can change the salt. And we have an example here. The salt improves the solubility. It gives you a very rapid, very fast rate of dissolution just because you have a different salt, right? Resulting in improved bioavailability. And this is example from the literature. And this were actually for the penicillin. Yeah, the penicillin. The penicillin, you have different kind of penicillin. Penicillin V, for example, it's a different salt than penicillin V calcium and penicillin V acid and sodium penicillin. So all these, they have different kind of rate, and of the solution, or actually in vivo, and the bioavailability is really a function of the salt you are using, right? From the top, which is the best, is the penicillin VK, and the worst was, or actually the slowest was, or the lower bioavailability was sodium penicillin, affecting the bioavailability. Here, this work we did at Roche during my time at Roche. When you have a free base, typically you would have like very low bioavailability. But when you have mesylate salt, if you use a mesylate salt, in that case for that particular medicine, just changing the salt from a base, it will give you better bioavailability. Another way of overcoming the issue of the bioavailability, overcoming the issue of low bioavailability or low AUC is the lipid. I love lipids. I work a lot in lipids. One of the approaches is basically self-emulsifying drug delivery system. Basically, what you have is you have a lipid system like soybean oil, something which is non-hydrophilic, something which is more hydrophobic, but it's a lipidic system. Also, another drug called Cabmol, which is all this kind of work we did at the time, and those are like a combination of a lipid system. Lipid means like fats. And also you have emulsifier like a PEG 400 or so, or a surfactant. And also, you could have like, an emulsion system. You can make an emulsion system and you will emulsify your drug. And the bottom line for all that you want to do, you would like to solubilize your medicine, make it a solution to give you maximum absorption. The drug will never give you, or your product will never have better better bioavailability unless you have the drug in solution. If the drug still have some particulates, you'll have problem. You have a problem. So you want to make sure that in this case, and many cases, that the drug is in solution. So the lipid plays a very important role, and the digestion also, because when you have a lipid, just like you have a fat, it's going to be digested. So if you are having a triglycerides or you have monoglycerides, lipids, it will make a difference. Okay, and if you have question, we can talk later about it. Particle size of the emulsion also. You may have a micro emulsion, or may have like a just typical emulsion. Micro emulsion obviously will give you better, kind of bioavailability. For this drug, for example, the in vitro release, if you can see, it's very interesting. In vitro release, just watch this one. The PEG 400 is give you the best actually in vitro dissolution. The self-emulsifying, which is what I'm trying to mention, the SED, we call it self-emulsifying drug delivery, give you number 2, that means slower dissolution. However, and the other two are slow. But if you look in vivo, which is most important, it's a different story. Why? Because when you have in vivo, you will have more enzymes and others which will digest the lipid. Digesting the lipid will lead to this. So you'll have self-emulsifying solution, give you far better relative bioavailability than the PEG 400 control. If you go back to the in vitro, it is flipped. The green one, which is this, and now it is number 2. The green comes here. So the in vivo, it behaves better than in vivo. What I'm trying to say in terms of bioavailability, as related to the self-emulsifying drug delivery system. So, and the rest of the other, the other two, wet milled spray and dried powder and the tablet didn't work well because see the bioavailability very low, which is like 35%, 17% compared to 389 for the self-emulsifying drug delivery system. Okay, and the reason is because we use the right lipidic system. So read about lipid. You guys, the students, your professor will teach you. This is a growing area. Although this has been for a while, but there are tons of lipids vehicles and technologies which you can use. You could use nano lipidic system. You can use self-emulsifying drug delivery system. You can use micro or actually micro emulsion lipid delivery system. There are so many of these I published. And in this and other people also. And if you have a question, again, you can always, be in touch with me. The other one we did already, this is in the market, something called Fortivase and HIV also. And this one we had two trials. We have a trial with just a regular capsule, which has a powder, and the other one also has the lipidic system. And you can see the difference. The AUC for the normal capsule containing powder is 297, what is actually double when we use it containing sets, when you have a lipid system on it. Okay, and the challenges were actually poor bioavailability, first pass effect, and the gut metabolism, cytochrome 450. All this could be an issue because of the nature of the GIT. But when you have the right system, a lipid system, the formulation, I can help you on that. I can send you some, your professor will do, obviously. You can improve the bioavailability due to the increased solubility and also modulation of the GI transport inside, because there are all these factors, and the right lipid system will help you to overcome all these issues of the poor bioavailability due to a first pass effect and liver transformation, etc. I'll take you now to the last one because of sake of time. I think you guys give me only 30 minutes, probably I passed it already, but I'll give you a quick overview of another technology we developed during my time at Roche, which is developing a stable amorphous system. Okay, and this like a patent already, we have it for this drug, which was very difficult to solubilize, very difficult, and oncology medicine, very difficult to solubilize. So basically the challenge is we have this oncology medicine, as I said, it has solubility which is very low, very sparingly soluble, less than 10 microgram per ml, which is nothing there. And very high low solubility. We tried lipid, which I mentioned to you earlier, it didn't work. So what else we could do? So what we did actually, I think, let's go back. Yeah, just so you could get the bottom line. Convert the crystalline form into amorphous. So we amorphosize the system. This is a growing technology, probably known by now, but during that time, at my time at Roche, 10 years ago or so, this was really growing, and I did a lot of work on it, on hot melt extrusion. Unfortunately, I might not have the time, but you could do hot melt extrusion, you could do amorphous by this way. So basically, you have the polymer, in this case, mesacrylic acid polymer, Hydrajet L100, maybe you're familiar with it. And then you have the crystalline drop, very low solubility, as I said, 10 microgram or less. The polymer drug together, you dissolve in organic solvent, could be methanol. I don't like methanol, you could go for ethanol. Okay, and then you quench it in the lab under cold acidified water, filter it, that means you give stress on it, washing it, drying it, you end up with a stable amorphous system. Why stable? Because you have the polymer and you see it here. Before I go to that, you have the polymer, you have the crystalline drug amorphous system created because you quenched. Somebody will say to me, how did you make it amorphous? Because you dislocated the crystalline. What is the crystalline material? It is well-defined structure of the crystal itself, right? What the amorphous does, or what this process does, it actually break all those kind of well structure and make it amorphous. Amorphous means it does not have well-structured system. It's an amorphous, right? So when you look under the microscope, or actually when you do DSTC, or you do like an X-ray, diffractometry, you see no peaks, okay? No peaks of a crystalline material, all right? So when we did the bioavailability testing, tremendous actually improvement in bioavailability compared to even nanosystem, compared to micronization, 800 over almost 900 compared to 2986. So it really does work well. And here it shows you, even you can have a control release because you have a polymer. Intermediate L100 is a polymer which can give you a control release kind of, attribute. And the amorphous system will be embedded in red here and locked within the polymer system. And that itself will make it stable for years. For 3 years it was. Emerging, last slide for me. I'm sorry for, maybe I took some time, but this I listed for you what is emerging actually now, what is coming, what is interested to us in the industry, being in the industry for the last almost 30 years. Nano-size system is growing. Libet will talk about it. Amorphous technologies, hot mic extrusion to give you amorphous, whatever, nasal, pulmonary, buccal, oral, fast disintegration dosage form, implant technology, needle-free injections, drug targeting, tumor targeting, biochemical triggered delivery system, nanotechnologies based system, and what we call drug reservoirs in microchips. So this is just what is going on in the industry, and you will share the slides, and each one of them will need a whole day. Formulation and discovery in conclusion, and discovery colleagues should closely work together, because you have discovery folks who are pharmacologists, medicinal chemists, you have formulators, and you have analytical folks. You have to work in a team because these are multidisciplinary team, which will be very helpful, to develop the clinical and also develop the clinical optimization of your lead, and also to de-risk your bioavailability. Because most of the issues when you are actually in discovery, which kills most of the good molecules, it's just because people, for a reason or another, cannot solubilize them, cannot make a good bioavailability. All the story which I mentioned, that can kill medicine, that can have, an issue. Our attempt or endeavor should be to select the molecule which is conventional. You develop it conventional. You don't want to make it complex. But unfortunately, sometimes in certain cases, because of the issues, because of the challenges we just talked about, classification 2 and 4, for example, you may need to do some kind of innovation, some kind of what we call it intervention of the formulation. So bottom line is formulation goal should be to develop. Most important for you in your head, you have to be as a student and working with great professor in this university, is to develop a robust product. Robust means what? That means it does not have stability issues, it does not have manufacturability issues, it does not have all what we talked about, bioavailability, etc., and also easy to make, probably cost effective, and also can go to patient in record time. So this is what we mean by robustness. If you do that, everybody will love you, and you'll have very good kind of future in the pharma industry in the United States and the great country of India and others. Okay, so with that, I thank you so much. Send question to me, Hashim Ahmed at Pop Council, where I actually work, because I'm the director of the R&D there, .org. We are actually a global organization across the globe. You are welcome to know more about the Population Council, and please send your question to me if you need more about this presentation, or if you need other related aspect. With that, I thank you so much, and I conclude by thanking you again for inviting me here today. I really enjoy seeing all of you across with that enthusiasm, and that will make this country, India, great as a pharmacy of the world, as we always say. Thank you guys so much.
Thank you so much, Doctor, and thanks so much for that informative presentation. We have a question from the chat box. Professor, can you please give us quick hits for protein peptide drug development from Waba, Amir Waba?
What was the question again? Say it again. I'm sorry, because I have some audio.
Can you please give us quick hits for protein or peptide drug development?
Yeah, definitely. Yeah, I think, yeah, protein and peptide, that if you guys invite me, I can devote a whole day for it, but definitely that's a different kind of situation. Basically, in the protein, you need to make sure that, obviously, again, make sure your structure of the protein and the sequence of the amino acids in the protein are well defined, and also you will have, the right kind of what do you call it, specification and also controls, because the protein is much more, complex, and also the downstream and the upstream process has to be well defined, just like any process. It's no different in that respect, but it's become more complex because of the characterization, the larger molecule, 40 or 50 or 100,000 kind of Dalton. So it's a little bit kind of more complex, and I can give you that. And if you invite me another day, I'll be more than honored to devote a whole presentation on protein and peptides. It's a growing field also. Maybe actually the rate of growth more than a small molecule now, but it's just like any other science. You need to understand the issue and also making sure you have the right controls. It's very important to understand the upstream process, which is actually, the infiltration and also the fermentation and all of those steps. And also, the downstream process when you conclude everything and making sure that you have the right protein. The sequence is very important, as I said. There are so many kind of technologies to, or actually, what do you call it, analytical methods, thousands of them to make sure the protein is well defined, structure-wise, sequence-wise, as well as stability-wise. Problem with stability is all the time because you're dealing with a protein. Storage of the protein has to be the right way, maybe minus 80 sometimes. So there are a lot of kind of different kind of challenges in the protein and peptides.
Thank you, doctor. And yeah, doctor, would you mind putting your email address in the chat box so that anyone would like to reach you out, they're going to reach you via email?
Yes, ma'am.
I have another question from Manoj Kumar.
Mm-hmm.
Can
Please
you
do.
please give us some names of drug to treat rheumatoid arthritis?
Mm-hmm. Okay, that's good. Rheumatoid arthritis. I think one of the drug, COX-2 inhibitors. Those are very good ones. Okay, Mexiclicam. I think that is the name. And also, even the ibuprofen is rheumatoid arthritis is a good one. Again, I can send you like more detail, but this top of my head, COX-2 inhibitors. Those are the latest, and even those non-steroidal. Non-steroidal anti-rheumatic drugs like ibuprofen in particular, and also what else? Yeah, I think there are so many of them, but non-steroidal anti-rheumatics is very important. COX-2 inhibitors. Yes. Yeah.
One more question, one last question. Can CCI technique be used to formulate herbal medicaments?
Herbal medicament. Yeah, what technique he said? I technique?
CCI.
CCI. The one I just sent, presented it?
Yeah, can it be used to formulate herbal medicaments?
Well, it depends on the herbal medicament itself, if it lends itself to that. So what you could do, if you have a herbal medicine and if you have an issue, the herbal medicine, the problem is the herbals, because they are like, you need to understand of course, what you have, the medicament itself, and also you need to control again that, the active ingredient. Yes, the answer is yes, but you have to know what you're doing, obviously, if that will be, what do you call it, amenable to it, if that process amenable to it. Most probably, it will be okay, but I need to know the issue itself with your herbal. It's because the herbal could have many issues. It might be just pharmacognostic kind of material, and you could fix it by a simple, like I said, conventional. But if it has a complex situation, if it has like what we just discussed, a crystalline material which is not controlled, keep changing into different kind of polymorphs, then what I presented is actually will be helpful to solve that problem. If it has another problem, like say, like we might have another technology, say like for lipid, maybe it can be helpful to have capsules, which will be, having those medicaments and the herbal. You may have also, hot milk extrusion, which is actually melting everything and then put them into a tablet and so on. So you could do different things depending on the issue itself. Depending on the challenge, the solution will come. So you know the challenge, understand your issues, like I mentioned, and then you have to put the remedy for it.
Thank you so much, doctor.
Sure.
Thank you so much once again for answering all the questions and for your presentation. Thank you so much. Welcome again, Dr. Amit.
Yeah. Thank you. Thanks for having me.
Our pleasure. Next, I would like to welcome our next speaker, Dr. J. Pillai, clinical pharmacist, Dignity Health, in Henderson, Nevada, United States. Welcome, sir. Good evening. Good evening to you, doctor.
Hello, Jojo.
Evening.
How is everybody doing?
We're good. We're good. I hope you're. Thank you so much for sharing your time with us, even this hour of the night.
Okay, that'll be my pleasure. Thank you so much for inviting me in this evening. It's about 10:30 or 11 o'clock in Vegas. So if I sound a little sleepy, if I look a little bit of lethargic, please understand.
Yes, we do. Thank you so much.
When I talked to Ramya Devi first, she contacted me asking me to do some presentation. When I looked at the name of the association, which is research-oriented pharmaceutical technology kind of focus, I told her, hey, it may not be that useful to the organization because I'm from a different specialty, which is a clinical pharmacy practice. And then she sent me a list of the topic they would be interested. And she identified hospital pharmacy and community pharmacy. And I told her, clinical pharmacy is nothing but hospital pharmacy. If you want me to speak about it, I'll be glad to do that. So that's
Yes,
how
please.
I came here. And if my topic is a little bit of an outlier, please take this as a dessert. I guess you will get the main course of the meal from Dr. Ahmed regarding formulation, structure, activity relationship, how to make the molecule soluble, how to increase the bioavailability, those kind of things he has already given you. And nanotechnology from Dr. Udupa. Those are the main course of the meal for your pharmaceutical appetite. Mine is going to be a little dessert. It may be a little bit out of your interest, but please bear with me. I will make it real short and sweet. Let me open up the screen here.
Sure.
Are you able to see my screen? Jojo,
Yes, doctor. Yes, doctor. It's very visible and we
okay.
can hear you clearly. You may proceed.
Am I clear in my
Yes, doctor.
speaking? Are you able to hear me properly?
Yes, doctor.
Excellent, excellent. So the topic for today is clinical pharmacy practice, which is actually a patient-centric approach practice in the US. What I'm going to be talking about is the practice that we follow here. It may or may not apply to India, or it may or may not be practiced in India, but it's something that one should know as to how we practice here in the US, which could be emulated in the future by the clinical pharmacist. My name is Jay Pillai, and I got a lot of degrees and names and all that. Please feel free to call me by my first name, Jay. I don't mind at all. Let's dive into the topic directly pretty quick. The objective of the webinar is to define clinical pharmacy practice. I will explain the role of a clinical pharmacist. And also I will go into the evolution of the clinical pharmacy itself based on evidence-based medicine. And I will give you a practice example from my own life experience. One of the patients that I came across in the hospital, and the doctor approached me for some help. I will explain that to put it all together so that it'll make some better understanding of the situation. And also, I will give you some examples of how to gather evidence-based information, and I'll give you some practice tools. If any one of you in the audience is interested in learning clinical pharmacy. Let's talk about the healthcare team for a second. Healthcare team usually focused on keeping the patient at the center, right? Patient comes first. And there are team that works on the patient's health, physicians, doctors, nurses, and transitional care people. And the patient is transferred from one floor to the other. For example, from intensive care unit to a different floor, there are some staffs available to transition the patient. And also we have the PT, OT, RT, physiotherapist, occupational therapist, and respiratory therapist. And then the case manager and social worker comes in. When the patient gets discharged, they'll make sure the patient is sent home properly. And what should the patient do to get back to the doctor and follow up? And of all these team, one of the important link that was missing is a clinical pharmacist all these years. And clinical pharmacy came into practice in the US back in the 1980s, 85, when I came to this country. And that's when it began in this country, and that practice began to grow. And after 30, 40 years, it is in full-fledged. And we are one of the essential personnel of the healthcare team. What does a clinical pharmacist do? What do the clinical pharmacists do? If you ask me that question, I would like to say we are the healthcare providers. We are the clinicians. We are part of the team. We also assume responsibility of medication therapy management. We are the primary source of evidence-based drug information. We are accountable for providing not only safe and effective medications, we make sure that it is also cost efficient. And what are the duties? These are my duties when I go to the hospital. This is what I do. This is not all inclusive of my responsibilities, but some of them, just to put it in a nutshell, I got here some bullet points. First, when I go to the hospital, what I do first, I do review the physician orders. The physicians enter the orders, drug therapy orders, lab orders, all the diagnostic procedures into the computer. And the medication list comes to my department. I look through his orders to make sure the drug is prescribed appropriately for the disease he is trying to treat. And also it is optimally dosed. I'll make sure there is no drug-to-drug interaction. There is no drug-disease interaction. For example, if the doctor prescribed Coumadin, warfarin as an anticoagulant, there should not be any drug interaction, preferably. I'll make sure there's no drug interaction. There's no drug-disease interaction either. The drug should not be incompatible with the condition of the patient. And also, I will do therapeutic drug monitoring. There are a lot of drugs that have narrow therapeutic index, just like digoxin, just like theophylline, just like aminoglycosides. They have narrow therapeutic index. Those drugs, when they are given, we have to make sure it does have the appropriate drug concentration so that the patient gets well. So they have the narrow therapeutic interactions. If it is too low, it's going to be subtherapeutic. If it is too high, it's going to be toxic. So we got to make sure it is in the right range of the therapeutic levels. And also I got to make sure the drug that is prescribed is cost effective. The doctor prescribed an expensive drug for a disease. And if I know a drug that is equally effective and it is less cost, then I'm supposed to make sure the doctor knows it. And we try to get the change to that cost-effective alternatives. When we do all these things appropriately, it does reduce the hospital stay of the patient. Ultimately, to reduce the overall healthcare cost. Finally, the goal is to improve patients' quality of life, which earns trust from the patient. And of course, that'll give you a professional satisfaction. The clinical pharmacy has evolved so much since the last 40, 45 years in the US. There are many different branches and board certifications. The first one is ambulatory care pharmacy. These pharmacies are specialized in outpatient pharmacies. Patient comes to the hospital, patient goes to the clinic, they get a prescription, and then they get it filled and go home. So they specialize in that aspect of the clinical pharmacy. There is cardiology pharmacy, like there are doctors, cardiologists. They do MBBS first, and then it's specialized cardiology, oncology, endocrinologists, those kinds of different specialties. In the same way, the clinical pharmacy has evolved into many different categories. Compounded sterile preparation pharmacy, critical care pharmacy, emergency medicine pharmacy, geriatric pharmacy for elderly people to make sure the drugs are dosed appropriately based on their age and their kidney function, nuclear pharmacy, nutritional support pharmacy, oncology pharmacy, pediatric pharmacy, pharmacotherapy. I do have the certification pharmacotherapy. If you see my titles, one of them is BCPS, Board Certified Pharmacotherapy Specialist. This is something like an internal medicine doctor. So I'm kind of a combination of all specialties put together. Psychiatric pharmacy, solid organ transplant pharmacy. So there is a pharmacist just allocated specialized when a patient has organ transplant to make sure the medication given to that patient is appropriate, prescribed correctly. He monitors, he makes sure a patient gets well. And if you learn the different board certifications of clinical pharmacy, you will go into the website, www.bpsweb.org. That'll give you a lot of information. And we practice primarily based on evidence-based medicine. We get the evidence-based medicine. How do you gather evidence-based information? We use the pyramid framework. If you look at the pyramid, like Dr. Ahmed mentioned, first, before you develop the drugs, right, you do animal research. In this case, the evidence quality increases as you go up the pyramid. First, animal research, then you publish case series and case reports and case control studies. These studies, they are not filtered because these studies are replete with errors, human errors, procedural errors, and also biases. So they're not filtered completely from those errors. When you go up the ladder, information gets filtered, and the information that you get from cohort studies, randomized clinical trials, systematic reviews, and meta-analysis, those literature gives you the filtered information, which has the best evidence. And we try to find information for the treatment purpose from the top of the pyramid, thereby We discuss with the doctor to make sure the medication prescribed is appropriate and it is cost effective, it works well. That's where we do an important role in the healthcare of the patient. Evidence-based practice tools. If you're interested, there are free evidence-based databases available. PubMed is one of the places it's available free. It is a website where you find a lot of clinical evidence-based studies, abstracts published by the National Library, National Institute of Health in Washington, DC. That's one of them is free, available for you. And also TRIP database is a good place to find some evidence-based clinical information. Cochrane Library is a combination of all these meta-analysis in the filtered information area. You can find it, which is very much used in this country. I'm sure some of you might have used it as well. And there are some for a fee website, ACP Journal Club, American College of Physicians, and also BMJ, the British Medical Journal, you guys would have come across, and also uptodate.com. I use a lot uptodate.com on my daily routine because this uptodate.com is actually up to date in publishing information, not only drug information, and also diagnostic procedures, other clinical information for the doctors. Doctors use it a lot. So when I look at this website, I'm able to see how the doctors think, and I'm able to provide accordingly the appropriate drug information with some evidence behind it. I would quote, according to uptodate.com, the recommendation is, these are the drugs that are appropriate for the disease you're trying to treat. And this is an example of my experience with one of the doctors that I came across on the floor. A physician requesting a clinical pharmacist, that's me, to treat MRSA infection. MRSA, I'm sure you're aware, it's the methicillin-resistant Staph aureus infection. It's a neurologist, he knows me well, and I'm on the floor, he comes to me, he's asking me, Jay, would you be able to help me treat this patient who has a MRSA infection in his wound? He's a neurologist, he is quite competent in his ability to treat neurological disorders, and he's not comfortable treating a wound infection. You could have actually asked an infectious disease doctor, but I was on the floor. He knows I'm a good clinical pharmacist. I think I am. He asked my help so that he doesn't have to speak to an infectious disease doctor, because that's going to add cost to the healthcare. And he has a patient, John Doe, who is 45 years old with penicillin allergy. His height is 72 inches, 68 kilograms, and his serum creatinine is 0.9 with a creatinine clearance of 90 mils per minute. The serum creatinine is very important because the drug we are going to prescribe is cleared renally. And the clearance tells me that this gentleman, 45 years old, has a reasonable kidney function, in fact, really good clearance. And the clinical pharmacist reviews the lab. I just looked at the lab because we have access to everything in the computer. His wound culture, of course, is growing MRSA infection. And the culture sensitivity also is displayed on the screen. And the MRSA that's growing is sensitive to vancomycin at a concentration of MIC, minimum inhibitory concentration of 1 microgram per ml. So I know he has a MRSA infection, and also I got the information that vancomycin is quite effective to this MRSA infection. And although he has a penicillin allergy, vancomycin is not a penicillin compound. So it's a quite inexpensive, safe drug, which will be effective in treating MRSA infection. And then I ordered vancomycin, 15 milligram per kilogram every 8 hours. The dose is based on his weight. He is 68 kilograms, right? It is 15 milligram per kilograms. So when you convert that into a dose, it goes a little more than 1,000 milligrams. So I rounded that off to 1 gram, 1,000 milligrams, because that's how the bottle injection comes in. So it should be dosed every 8 hours based on his creatinine clearance. If his creatinine clearance is compromised, if it is 40 or 50, this dose Q8 is inappropriate. I got to cut it down to probably Q12. Then I have to order a lab myself to draw the trough, because these drugs are effective based on certain concentration. So we have to draw the trough level. Nowadays, the vancomycin is also measured by AUC, area under the curve. But until very recently, we used to draw the trough to make sure the levels are correct. It is therapeutic, because if it is too low, then I have to increase the dose. If the trough is high, then I have to reduce the dose. That's how the lab comes in. So I am able to prescribe the antibiotic. I'm able to order the labs to monitor the patient. And I got the authority in the hospital for that. And then I monitor the patient. I make sure I look at the lab and adjust the dose if necessary. And this dose was appropriate. When I looked at the trough, it was correct. It was within the range. And then on the 7th day, the wound culture shows no growth. It's completely clear. The vancomycin eliminated the infection completely. And I reported to Dr. Selko, the neurologist. He was extremely happy. Patient went home. So this is one of the examples where the clinical pharmacist plays a key role in assisting the physician where his expertise is not sufficient. And of course, we guys play an important, crucial role in the important link in the healthcare chain, in the healthcare team. That completes my presentation of the dessert. Hope you get something out of that. If you have any questions, I'll be glad to answer. If not, you can send your questions to the email address j.pillai@gmail.com. I'll be glad to answer any of those questions. And I think I see Dr. Udupa in the picture. Is that you, sir? Yes, he is. Yeah, I met you in Manipal with B.R. Shetty. You might recall that. Nice to see you. Thank you so much for the opportunity given. And it's getting late. I am going to leave the door open for Udupa to come in and entertain you, your meal course of the meal. Thank you so very much. Jojo, any questions from the audience? Hey, Jojo, I'm done. Is there some technical difficulty? I think Dr. Udupa has to unmute. It appears he is not put in as a co-host.
Thank you. So I really do apologize. I wasn't able to unmute myself earlier. Doctor, thank you so much for that presentation. It was very informative. And yes, anyone would like to ask a question to Dr. Pillai?
I think, Jojo, one of the gentlemen put in a question in the chat. Some of the medications for rheumatoid arthritis.
Yes.
RA.
Oh yeah. What can you give?
I think it
that
was
can be
Manoj
given?
Kumar. For rheumatoid arthritis, I might be able to give some, better information coming from the hospital, clinical pharmacy practice. The mainstay of drug therapy for rheumatoid arthritis are simple, non-steroidal anti-inflammatory drugs, NSAIDs, starting with simple ibuprofen, naproxen, or meloxicam, those kinds of drugs, diclofenac. And also you have to, first of all, identify whether it is a rheumatoid arthritis or an osteoarthritis. If it is rheumatoid arthritis, the mainstay is the non-steroidal anti-inflammatory drugs. And if those drugs are not effective, then the next step would be some of the MABs, monoclonal antibodies, such as Remicade, infliximab. Those are all, IV infusions. One of the rheumatologists examine, and it is given, I believe, based on the type of disease and severity, given once a week or twice a week or once a month. Those are MABs comes second. Those are the approach when treating rheumatoid arthritis. Hope that's helpful.
Thank you so much, doctor. Thank you so much once again, and it was our pleasure to have you here with us tonight in your time. Thank you so much for sparing with your time with us. Thank you so much.
Thank you all.
Now I would like to invite our next speaker, Dr. Udupa, Research Director, Shri, Dharmasthala, Manjunatheshwara University, or SDM College of Medical Sciences and Hospital Campus, Karnataka, India. Good afternoon, sir. Good noon, sir.
So, good afternoon to APR organizing committee and previous speakers. They have given a lot of technological inputs and through light, and they have introduced this particular topic. And to continue, because my theme and topic is future scope of nano and biotechnology. So next slide. Yeah. So there is that innumerable drug delivery systems are available for drug delivery into our body for treating various diseases. They are like oral control drug delivery systems, parenteral injectable control systems, nasal and pulmonary drug delivery systems. The first and second speakers have mentioned about this, transdermal drug delivery systems, nanotechnology based systems, including nanoparticles, dendrimers, and liposomes as carriers. And most of these nanosystems are target delivery systems. Next. And there are a lot of oral control drug delivery systems which are available in market. They are in the form of beads in the tablet, like intestinal protective drug absorption system, multi-particulates and mini tablets with the programmable oral systems like ProDOS and SODOS, that is spheroidal oral drug absorption systems, and sugar spheres containing rate controlling membrane like diffucaps, which are the most earliest, about 3-4 decades back. This control drug delivery in the capsules they are available. Then urine mini taps. Then beads, multi-particulates like macrocrap, Orbexa technology, flash tap technology. These are all the technology of our oral drug delivery systems, which are already marketed, commercially viable products. Next. Then coming to like different approaches for oral drug delivery system. One of the the latest development is osmotic pump tablet. It may be hydrodynamically balanced capsules where with drug core and the orifice where the drug is released from the external environment of gastric or intestinal water. It will push there is a push layer which will push the drug from the orifice through, and a semi-permeable membrane will hold this, and the semi-permeable membrane will help for penetration of the external water like that. So the drug release can be properly with the hydrodynamic balance capsules. It can be easily through diffusion. So it can be, and diffusion and erosion can help to release the drug. This is the technology already available and popular. Several preparations are available based on this technology. Next. Then coming to next slide, please. Yeah. So under parental control drug delivery system, which is a very ideal route for the emergency with the fast onset of action, and it is useful when drugs are inactivated in gastrointestinal tract, and even for less bioavailable drug. So major routes for parental control drug delivery systems are intravenous, subcutaneous, and intramuscular. The novel parental control drug delivery systems are in the form of implants, microspheres, nanoparticles, liposomes, microemulsions, and nanoemulsions. Next, please. Then mucosal drug delivery. The mucosal surfaces are considered as potential absorption sites for drug administration. Drug is directly transported through mucosa to reach to the systemic circulation. The mucosal linings of nasal, vaginal, rectal, ocular, and oral cavity offers the transmucosal absorption of the drug. The nasal mucosa, which is present in the olfactory region, is specifically used for delivering the drug from the nasal cavity to the brain, to the central nervous system. The novel drug delivery systems like patches, tablets, thermosensitive gels, pH-sensitive gels, nanogels, microgels, they are useful for mucosal drug delivery system. Next. Then coming to the transdermal drug delivery, it is available in the form of through the skin to the systemic circulation as transdermal patches, as nanogels, microneedles, nanoneedles, and the delivery is through for the transdermal. There are 3 important techniques like iontophoresis, electrophoresis, and sonophoresis. That is for increasing the permeation. We are using dendrimers as skin permeation enhancers for several drugs such as 5-uracil. Transdermal drug delivery patches, they look like this, which is on the hand or on our chest, and through iontophoresis, sonophoresis, or microneedles, we can enhance the permeation. Next. The targeted drug delivery systems, they imply selective and effective localization of drugs to the targeted areas in therapeutic concentration. It restricts access of drugs to normal cellular lining, minimizing the toxic effects, maximizing the therapeutic index, and increasing the patient compliance. Then there is a significant reduction in drug toxicity, reduction of the drug dose, and increased treatment therapeutic efficacy. Next. The approaches for drug targeting, either passive targeting or active targeting. The various factors for passive targeting are pathophysiological factors like inflammation, infection, EPR effect. Then physicochemical factors like size of the drug molecule, molecular weight of the drug molecule, then anatomical opportunities like catheterization, direct injection. Then there are chemical approaches like prodrugs and chemical drug delivery system. For active targeting, the main biochemical targets are organs or cellular targeting or organelle targeting or intracellular targeting. Using physical or external stimuli like ultrasound and magnetic field, either electrophoresis or sonification, etc., we can enhance the active targeting. Then general approaches like a physical approach, it is known as also magic gun or magic missile, targeting to mononuclear phagocytic system, targeting to the pulmonary region, extravascular delivery, mucosal delivery of the antigens, then magnetic approach. The biological approach is known as magic bullet approach. It is by lectin as targeting agent, or using folate receptors, or using low molecular weight proteins for renal targeting. And also retrograde transport also is possible. The chemical approach, prodrug approach also I also mentioned. Various nanotechnology based drug delivery systems can be used for targeting drug to the tissues or cells. And I will be explaining some of those systems now. Next. Yeah. There are various marketed products for targeted drug delivery, like Ambizome. This is a liposomal amphotericin, one of the most, about 20 to 30 years back, Gilead Sciences, they introduced this. It is used for fungal and protozoal infection. Then deposit, this is liposomal cytarabine, which is introduced by Pfizer and is used for malignant lymphomatous meningitis. Then Myoset, which is liposomal doxorubicin, which is marketed by Genius Pharma, which is used for metastatic breast cancer. And also Doxil, like liposomal pegylated doxorubicin, which is again introduced by Orthobiotech and Shri Flow Company. Then Abraxane, it is a very the breakthrough technology by Pfizer and Abraxane Biosciences. It is paclitaxel nanoparticles for metastatic breast cancer. A lot of cost reduction and therapeutic efficacy improvement is there. Then Diprivan, which is using propofol liposomes by Zeneca Pharma for anesthetic this delivery. Next. Nanoparticle systems are potential drug delivery systems for poorly soluble, poorly absorbed and labile substances. Nanoparticle systems are used for diagnosis, for therapeutic treatment, and also for the drug and gene delivery, nutraceuticals and cosmeceuticals also. Nanoparticle systems are quite useful. And in producing improved biocompatible materials also, nanoparticles are a lot of breakthrough. Next. Then different nanosystems are represented pictorially here, like nanosphere, nanocapsules. And there is a polymeric missile with the hydrophobic core and the core and outside membrane-like thing, and the pictorially liposome with the aqueous core and then lipid bilayer, which can be further that second layer can be pegylated or conjugated with the antibody, and also ligand conjugates like zinc sulfide capped, that is CDS. This CDS is also there. Ligand conjugate is also there. Then the latest technology in this area is fourth generation dendrimers with the, and also ferrofluid conjugated to the ligand is also possible, because it's for the magnetic microspheres and nanoparticles, this is being adopted. Next. Then nanoparticles, they are having like nanospheres with the polymeric matrix, with the polymeric membrane and oily or aqueous core, and in the usually less than 200 nanometers are known as nanospheres, and nanosphere DNA complex with the electronic microscope we can magnify and show like this. It looks like this. Next. Yeah, the surface properties of nanoparticles, that is for modification and improvement of the enhancement of the absorption of the drug, the protein absorption and phagocytosis of nanoparticles. The methods of surface modifications are pegylation or polyethylene oxide coated nanoparticles, or coating with the negatively charged poloxamine and poloxamer nanoparticles. Even cyclodextrin and carbohydrate coated nanoparticles are being designed. Polysorbate coated, that is non-ionic surfactant nanoparticles, they have helped to penetrate blood-brain barrier for nasal delivery. This is for one of the latest technologies. And then pegylated nanoparticles, they decrease the hydrophobicity and reduce the toxicity. They prevent the oxidation and avoid the reticuloendothelial system uptake. That's why they improve the therapeutic availability and the enhancement of the therapeutic effect. Next. Then nanoparticles are very much used in cancer therapy. The conventional nanoparticles are rapidly oxidized and massively cleared by the fixed macrophages of the reticuloendothelial organs. The conventional nanoparticles can be enhanced to anti-cancer drug efficacy, for which is limited to targeting tumors at the level of reticuloendothelial system organs only. Whereas stealth particles, which are invisible to macrophages, like pegylated, they are known as stealth particles. They increase the anti-tumor efficacy, reduce the systemic side effects, selectively deliver the oligonucleotides to the tumor cells. They are characterized by the prolonged half-life. They are small size, like 100 nanometer, and hydrophilic surface reduce oxidation and clearance by the macrophages. Next. The nanoparticles, like the long circulating nanoparticles for cancer treatment, in the healthy endothelium and the tumor cell, the interstitium, the stealth nanoparticles, how they circulate, and how they remain for long times, get circulated and release the drug for the prolonged time. That's how, and it is quite stable. That's how it shows the maximum therapeutic effect. Next. Then pictorially, like I told, nasal to the nose to the brain. Actually, free drug, you'll see that in the semi-permeable on the other side, the amount is, whereas as nanoparticle, it has increased retention in capillaries combined with the absorption that leads to higher concentration gradient, solubilization of the endothelial cell membrane lipids, which leads to enhanced drug permeability. And nanoparticles open the tight junction between the endothelial cells because of the surfactant and other permeation enhancing effect. Nanoparticles may be endocytosed or transcytosed by the endothelial cells, like polysorbate, that is Q80, that non-ionic surfactant. It can inhibit the efflux system, PGP system, the pharmacological system. Yeah, next. Then this is how the nasal drug delivery to the brain it works. Like nose, it can be cleared, whereas olfactory region, because of the high vasculature effect, it reaches to the through the blood to the brain tissue and cerebrospinal fluid. The blood slowly elimination starts. That's why nasal drug delivery is more effective. About 70 to 80% bioavailability. Systemic, this one, there is at least 100% intravenous infusion, etc. Whereas nasal drug delivery, we can achieve 70 to 80%. That is the best alternate route. And also non-invasive therapy. Yeah, next. So there is another system known as micellar system, similar to nanoionic surfactant. It is a colloidal carrier system used for targeting drugs that have low aqueous solubility, and also it has amphiphilic molecules. Micelles have hydrophilic shell exhibiting long circulating time and accumulation at the target site. It has high loading capacity, and attachment of the homing device is also possible. And physical ability of the targeting using stimuli responsive polymers, like the stimulus is like pH or electromagnetic field or temperature difference, high temperature. The additional cross-linking in the core or cell can lead to novel nanostructure with different drug delivery properties. Next, please. Then dendrimers, which is the latest development which is taking place, and it is a small nanostructured compound synthesized by the continuous additions of layers of branching groups. The molecular structure has the form of tree with many branches. The many types are Pharma, that is the polymer like polyaryl ether or polyester or PPI or tectodendrimer or amphiphilic dendrimer or peptide dendrimer. They serve as nano device for delivery of therapeuticals, like therapeuticals like oral, and they can be delivered through various routes like oral, transdermal, ocular, parenteral, etc. The attractive candidates for the drug is for using dendrimers are antivirals, anti-cancer agents, vaccines, gene, that is siRNA, the DNA, and also diagnostic agents. Next. And dendrimers, they are represented like this. They are highly branched with the core moiety and globular structure. Then there are internal cavity and also branching units. And they are closely packed surface groups. The core is GO, stepwise addition of the monomer, the first unit, second G1, G2, G3. So this siRNA and DNA can be the charge association can take place, and then endocytosis can happen with the dendrimer as carrier. Next. Then application of this dendrimer as carrier is there is a lot of advantage with the solubility enhancement. It is possible using cyclodextrin micelles, and ionic hydrophobic interactions at hydrogen bonding is possible. Then generation, concentration, pH, temperature, terminal functionality, all these are very important factors. The pegylization maximizes drug loading and solubility. For example, pyrene, artemether, niclosamide, ibuprofen, all these drugs can be delivered through the dendrimers. Next. Yeah, the solubility environment, the previous one. Yeah. The solubility environment and all these, yeah, next, artemeter, already I have told, next. Yeah, dendrimers as drug delivery systems with the micelles, cyclodextrin and palm dendrimer. It looks like the structurally they are represented like this. Next. Then dendrimers as carriers. Second one is oral drug delivery. The advantage with dendrimer is the controlled release, increased solubility and stability of the drug, more gastrointestinal residence time, and better absorption potential. The dendrimers can penetrate gastrointestinal membrane and enhance the gastrointestinal mucosal penetration of the drug, and conjugation of lipid chain decreases the cytotoxicity. The examples of the drugs which can be delivered orally as dendrimers are naproxen, propranolol, and 5-fluorouracil. Yeah, next. Then transdermal drug delivery. Dendrimers improve drug solubility, stability, and act as penetration enhancer for ketoprofen, difensal, indomethacin, nifedipine, 5-fluorouracil, etc. Intratumoral and intravenous, intrapulmonary routes are also possible for rapid onset and most direct route, and also for intramural useful anti-cancer drugs like glucosamine, doxorubicin, methotrexate, and fluprophen. Next. Then ocular drug delivery, like pilocarpine by palm dendrimers using COH or OH surface groups. The advantage is slow release of the drug from the interior cavities, bioadhesive property of the palm dendrimer, and other drugs like glucosamine, glucosamine 6-sulfate also can be delivered through the oral route. Pulmonary drug delivery is another application. where PAM and dendrimer showed increase in pulmonary absorption of anticoagulation agent enoxaparin. Yeah. Then targeted drug delivery, already I mentioned passive targeting using dendrimer, where radius is increased by pegylation. Equalization of dendrimer where EPR effect is possible. Then active targeting tumor cells to the not to the target areas like your lung and spleen, to target less vascularized or inaccessible tissues, then targeting ligands like carbohydrates, FA, monoclonal antibody peptides, which are conjugated. Next. Then the control release of the drugs, drug entrapment ability of dendrimer, ketoprofen, with Pharma, and methotrexate is possible through pegylated 2000 Pharma. Miscellaneous routes like rectal, vaginal, nasal routes are also possible. Dendrimer based vaginal microbicide vivo gel is already available in the market. Next. Then dendrimer has some of the anionic dendrimers exhibit antiviral activity. Like dendrimers bind multivalently to cell surface or to viral components. They inhibit the infection of HIV, RSV, HSV, etc. Vivogel, marketed by Star Pharma. It is a commercial vaginal dendrimer formulation against HIV. Some cationic dendrimers for lysine conjugated microbicides. They interact with membranes, negative charged membrane, leading to the lysis of the bacterial cells. Next. Yeah, there are specialized lipid-based nanosystems like microemulsions and nanoemulsions. Microemulsions, they are transparent in nature, they are homogeneous, thermodynamically stable, they are dispersions of oil and water. The size is between 10 to 140 nanometer. They can be stabilized by mixture of surfactants and co-surfactants. Then SNES, they are isotropic mixtures of oil, surfactant, and co-surfactants. They are spontaneously formed and will give rise to oil in water type nanoemulsion upon mixing with the GIT fluid. The size is about 50 nanometers. Next. Then microemulsion, then ethosomes. They are soft, malleable vascular carriers consisting mainly of phospholipids, high concentration of ethanol and water. They possess higher skin permeation than liposomes. Then there are transferosomes. These are highly deformable lipid vesicles, achieve high transferable delivery of the drugs. Then cochleates. They are solid particulates composed of large continuous lipid bilayer sheets which have been rolled up in a spiral structure without internal aqueous phase. Next. Next, please. Yeah, pharmacosomes. Actually, Peter Spicer, as soon as non-ionic surfactants are even earlier to that, for targeting like anti-cancer drugs, anti-infective agents, the pharmacosomes are discovered. These have, they are just drugs tagged with fatty acid derivatives. They are amphiphilic lipoidic vascular carriers composed of phospholipid complexes. The drug is bound to the phospholipid via covalent electrostatic or hydrogen bonding. They exist either as ultrafine, micellar, or hexagonal aggregates depending on the basis of their chemical structure. They are used for ophthalmic delirium drugs. Then archaeosomes, liposomes fabricated using polar ether lipids from archaea or using synthetic lipids which possess peculiar properties of ether lipids. They are more stable than liposomes. Next. Next, yeah. Next, please. Next, please. Yeah. Solid lipid nanoparticles. They are known as SLNS. They are controlled drug delivery systems with the bioavailability enhancement property, improvement of distribution and drug targeting. They are better than micro and nano emulsions and polymeric nanoparticles. Then there are nanostructural lipid carriers. These nanostructural NLCs have advantages over SLNS as they have higher drug loading capacity and reduction in drug expulsion. They are formulated using mixture of solid lipid and liquid lipid. Next. Then lipid nanocapsules, they possess hybrid framework in between liposomes and polymeric nanoparticles, improve oral bioavailability of poorly soluble drugs. Lipospheres, lipid microspheres and lipid nanospheres, they are similar to lipid nanocapsules, but formulation in matrix form. They improve the oral bioavailability of poorly soluble drugs. Next. So now I'll give some examples based on our laboratory work in Manipal Academy of Higher Education with some examples of papers published in international journals. So one of the applications is biological nano device based on dendrimers are being developed with the potential with the apoptosis sensor imaging for X-ray, MRI, and also cause for the disease also can be discovered. Cancer targeting is possible, therapeutic effect is possible. They recognize cancer cells, diagnose the cause of the cancer, deliver the drug to the target, report the location of the tumor, report the outcome of the therapy. Cancer death also can be demonstrated. Next. Yeah, there are various technological challenges in this technology, like drug release in specific areas in a controlled manner, biocompatible and biodegradable material for construction, then active targeting, on command, smart delivery, intelligent device, bioresponsive trigger system, circulatory system, interacting in the body, smart delivery, then improve devices like implants, nanochips for nanoparticle release, or multi-drug delivery chips, tissue engineering and delivery of peptide protein, toxicity issues, combined therapy and medical imaging, cell and gene targeting system, better disease markers in terms of sensitivity and specificity. So these are all the technological challenges, and for all pharmaceutical biotechnologies, it is a very important area. A lot of grants, funding agency, industries are there to collaborate, help us. Next. Then drug release specific areas, controlled manner. Yeah, next please. Yeah, the future perspective is for the development of affordable novel drug delivery systems are polymer therapeutics, which is an expanding and credible role for specific targeting. Following approach, like differences between cancerous cells and normal cells, insensitive to negative growth regulation and anti-growth signals, angiogenesis, metastasis, development novel, sophisticated, multifunctional system which can recognize cancer cells, deliver drug to the target tissue, and aid in reporting outcome of the therapy. As nanosystems become more complex, ability to assess and control reproducible uniformity of manufacturing represents great risk and challenge. The subtle changes in complex structures and compositions may have dramatic effects on safety and efficacy. Next. Lab findings, some examples like design and develop nanoparticle systems for the treatment of brain cancer by nasal route, where the polymeric nanoparticles in the form of nanosuspension or SLNR of anti-cancer drugs like using carriers like polylactic glycolic acid, polycaprolactone, and polylactic acid. And we have studied this with the FTIR and differential scanning calorimetry and size optimization. Then the polymer dispersive index, then the zeta potential, drug EPR effect, and nanoparticles show sustained release profile. All these demonstrated in vitro anti-cancer studies. Human brain cancers have revealed that enhanced and prolonged cytotoxic due to enhanced permeability. Next, to prove all these facts, we have a lot of publications, international publications. Next, by our research group. That is yeah. Next, please. We change the slide. Yeah. So we have used in vivo pharmacokinetic studies in animal models, where we through nasal route we found more accumulation drug in brain via nasal route that versus intravenous route. Drug targeting index for the non-particulate formulations were significantly higher as compared to pure drug. Nose to brain pathway draws attention to the treatment of brain cancer. Next. So another drug is calcium channel blocker, which is a typical dehydropyridine loaded into nano proliposome. This is the molecules lacanadepine. We are successful at in formulating lacanadepine nano proliposomes by modified thin film hydration technique using a cryoprotectant solution as film hydration medium. The novel formula showed a particle size of 174.7 nanometers and entrapment efficiency of about 86% was demonstrated. It appreciable extended drug release pattern, and in vivo pharmacokinetic studies has shown improvement in pharmacokinetic efficacy, and we saw that compared to the plain drug, it has enhanced about 3 times increase in overall bioavailability. Next. So this is how the design, it goes, the machineries and the pilot manufacturing of lacanadepine, with where lacanadepine lipid and cholesterol, they are mixed with the chloroform, and they are there is a rotating flask system is there. Downside at about 15,000 psi for about 10 cycles at 15,000 rpm 10, and deep freezing is required, drying at minus 40 degrees centigrade to minus 50 degrees centigrade with vacuum, and to remove, and after removing the residual organic through the vacuum pump, that's how the nanoproliposome of lacanadepine are prepared. Next. Next, please. Yeah. So these are some of our international publication elsewhere, like Science Direct, a novel nanoproliposomes of lacanadepine, the developing vitro and proclinical studies to support is effective in hypertension therapy. It is already being published. Next. Another brain targeting nanoparticle systems for treating Alzheimer's disease. With all some PhD studies, the person who worked with me now is in Korea, working for a company where donepezil and curcumin are being used, an irreversible progressive brain disease that slowly destroys memory and thinking skills. The amyloid beta plaques, dense deposit of protein and cellular material that accumulates outside around cells, and neurofibrillary tangles, twisted fiber that build up inside the nerve cells. It is all pictorially represented here. How the brain targeting we have demonstrated. Next. Next, please. Yeah. The brain targeting of nanoparticle systems is produced with the higher brain ACH inhibition than the plain drug combinations. Nanoparticle treatment for 7 days produced clear evidence that the drug combination showed significant improvement in various pathological conditions of the disease. The histopathological evaluation showed that the drug combination nanoparticles reverses the toxicity produced by the neurotoxin. The plasma concentration of the drug was observed in the quantifiable level, which could be due to long circulating nature of the nanoparticle. The improved pathological condition, reduced organ toxicity also may be due to precise brain targeting ability of the nanoparticles. Next. Now, this is how we have demonstrated the better bioavailability and therapeutic efficacy. Like the prepared nanoparticle and drug combinations are biologically and environmentally combinationally safe. These drugs protected the brain and the organs from ALCL3 induced toxicity and safely delivered the drug to the brain in a sustained manner. So, donepezil, curcumin, and combination of ACH inhibition. So, in vitro drug release and ACH inhibition showed that the nanoparticles produced a sustained release profile throughout the experimental period. Yeah, next. For about 160 hours. Then brain targeting nanoparticle systems we have clearly studied, and the disease control and nanoparticle treatment. We have seen that the nanoparticle reduce the brain toxicity induced by the neurotoxin and treated the Alzheimer's disease model. It is tried in the rat model. Next. Yeah. So this is how the brain distribution studies of nanoparticles is studied for pegylated, the PLGA, then with the nanoparticle system and pegylated nanoparticles, and the plain and surface modified nanoparticles significantly distributed to the brain and effectively treated the Alzheimer's disease model rats. Next. Next. Yeah. So this is how the brain distribution studies are represented. The percent ACT inhibition on the brain biodistribution is being represented here. Next, please. So artificial cells nanomedicine biotechnology is one journal where the bioactive PLGA curcumin microparticle embedded chitosan scaffold. I mentioned Muthukumar is there with the is in now Korea. He worked one of his PhD work which is published in one of the international journal. Next. Yeah, brain targeted nanoparticle systems. A journal of microencapsulation also published one of our paper. Same again Muthukumar's paper. And in vitro biocompatible release of curcumin from curcumin microcomplex loaded chitosan scaffold. Yeah, next. And this is how oral dispersion film containing drug nanoparticle lactodapine is being represented. Next. The electron microscopic picture of the same. Next. And in vivo, ex vivo formulation studies through porcine, buccal mucosa, and goat nasal mucosa we have demonstrated here. And clearly, higher formation of drug with the nano pharmaceutical gel is being demonstrated here. Next. So here are all pharmacokinetic data. When we compare oral and nasal, all Tmax, Cmax, AUC, the relative bioavailability, everything is almost multiplied several, almost double time. Greater pharmacokinetic parameters were observed by nanoparticle dosage forms in a comparison with pure drug, justifying better therapeutic efficacy. Yeah, next. These are all animal studies. Next. Next, please. Yeah. So another European Journal of Pharmaceutics and Biopharmaceutics, where development of hard dissolved oral films containing lactodapine nanoparticles. So my all co-authors and like Pratik Jain is in Canada, Ankita, she's in Bombay working for a company, and then all these are all PhDs, and my colleague Dr. Udupa is a biotechnology, so we collaborated, and Manipal group has published this paper based on our good work. Yeah, next. Yeah, so there are self-regulated drug delivery systems with the closed loop systems, feedback regulated drug delivery system with the pH sensitive showing shrinking properties using polymeric acidic functional group like SO3H, COH, which swell in basic pH, and basic groups like NH2 which swell in acidic pH. Next. They are self-regulated drug delivery systems. Next. Then thermoresponsive drug delivery. The polymers with least critical solution temperature, so negative or inverse temperature dependent behavior, they are insoluble as temperature increases. They shrink as the temperature increases above the LCST. LCST of polymer is low if the polymer chain contains lesser hydrophobic groups, more hydrophilic moieties enhance dissolution, and they show the burst release. The balance between hydrophobic and hydrophilic moieties is important in thermosensitive hydrogels. Polymorphs can be used for this designing thermoresponsive drug delivery systems. Next. So competitive binding delivery with the pH sensitive hydrogel containing glucose oxidase, insulin solution, glucose oxidase, and breaks into gluconic acid, which decreases the pH, and swelling of the membrane takes place, and insulin is released through the swollen membrane. Insulin is derived and closed loop insulin. So, like glucose response enzyme utilizing system is being shown here, coated with hydrophilic nylon to form micro capsule and increase blood glucose level. This was insulin from concanaval vanillin complex. This is being patented, and actually Biocon is trying to deliver this drug, but still there is some technical difficulties. Hope we will see that sometime. It will be very for the hyperglycemic patients for the insulin delivery. It will be a very good system. Next. So this is how the animal studies are being demonstrated where glucose intake for hyperglycemia, closed loop insulin release take place, and then normal glycemia, and then basal insulin release. Closed loop insulin release of self-regulatory system is being represented in this particular picture and cycle. Next. So system utilizing antibodies like naltrexone, the matrix system, drug release is triggered by morphine in blood. Drug is dispersed in polymer matrix coated with lipid layer that prevents water entry to the matrix. Delivery system is placed in dialysis bag which contains a lipase conjugated to morphine anti-morphine complex. Then inflammation-induced drug delivery system is also possible. This approach is used for inflammations like for treating rheumatoid arthritis. Drug-loaded lipid microscopies are dispersed into biodegradable matrices of cross-linked hyaluronic acid and injected at inflammatory site. It gets specifically degraded by hydroxyl radicals produced from inflammation responsive cells during inflammation. Yeah, next. Yeah, so that's how I have shared some of our examples, and there is a lot of scope for nano and technological scope is there in future, and all scientists, our industries and government agencies and the organization like APR, because whatever we work, we should be showcased to the people, all the scientists and other industries, etc. So this type of platform. So I thank very much for APR and all the full team and my other co-speakers who helped us for conducting our today's program. So thank you very much. If there are any questions, I'll try to, and either my mail ID and all are there, and I'll be either explain or otherwise I'll be sharing those reply. Because of the time shortage, I don't know how much time is there. Yeah, please, any questions are there? I'm very pleased to clarify and answer. Thank you. Thank you very much. ಅವರು ಇದು ಮಾಡಲಿಲ್ಲ, ಅನ್ಮ್ಯೂಟ್ ಮಾಡಲಿಲ್ಲ. ವಿಶ್ವಾಸ ಬಂತು. Yes, madam, you have to unmute. Is there any question in the chat box? Can you unmute? Jojo, yeah. Not able to listen to you.
Hello, thank you so much. He wasn't able to unmute. All right, yes, and thank you so much, doctor, for that very informative presentation. Anyone would like to ask a question? We have a question with
It is the best and most suitable drug delivery system. Yeah.
Yes.
Nose
Which
to
is
the
the best
brain.
and most suitable drug delivery system for nose to brain delivery?
Yeah, that is as already mentioned, the either dendrimer or the liposome encapsulated pegylated with the surfactant system in the form of a micelle is very good, or even in the as a gel based system to the, if we can apply to the nostrils, we will be able to do that, because we have already demonstrated and some paper we tried pilocarpine and some model drugs, and very good, and even insulin can be delivered through the nose. So some experiments are done, and some data I have already shared. So that's how we can try.
Any other questions?
So yeah,
I think none. All right, I've already shared the YouTube live coverage of this webinar. You can review our webinar on that link address here. I'm gonna be pasting it again. There you go.
Yes, yeah.
And again, I would like to thank um, BioLeagues and United Innovators as our partners for today's webinar. Are also our speakers, Dr. Ahmed, Dr. Pillai, and Dr. Dupa. Thank you so much. All right, now on behalf of the Association of Pharmaceutical Research, or APR, and DY Patil University, I would like to express my gratitude to all esteemed delegates of this webinar for their presence and contribution to make this webinar more informative and interesting session. I extend my gratitude to our keynote speakers and DY Patil University to taking their time from from their busy schedule to grace this event. Again, thank you so much, all participants and all the listeners of our webinar today. Once again, this is your host, Jojo Mehoy. Good evening, good morning, good afternoon, everyone.
Thank you, Jojo.